All right, so we have the recording here. Welcome, Braden. Welcome, Karis. Welcome, Gabriel. Welcome, Gavin. Welcome, Grace. Welcome, Marshall. Welcome, Meredith. Welcome, Shelby. Welcome, Isabel. So thank you for reminding me that I hadn't graded the Chapter 5 tests. Those should be graded now. And they are open to corrections as per the usual method. And today we are going to cover pages 221 to 226 and kind of have that be at the end. We're almost here at the end of Chapter 7. So we're really going to go in depth onto chemical synapses. Chemical synapses. And remember that one time when we were like joking about how various drugs alter the brain, etc. We're going to take a look at how it is exactly that these addictive substances influence the brain, because they target the chemical synapse. So please turn to page 223 and we'll take a look at that today. Let's pray. Lord, we thank you for this day that you have made. We ask that in your mercy you would continue to be with any brothers and sisters in Christ who are firefighters or health professionals or other emergency service folks over in California. We ask that in your mercy you would enable them to continue serving and save as many people as they may be able to. Father, we ask that you would be with your church as they help people rebuild after the devastating fires. We ask this in your Son, Jesus' name. Amen. Okay, so on page 223, there is an image of the synapse for us. And I'm going to put this up here on the board, and we are going to look at how neurons talk to each other. And we've seen a little bit of this in the neuromuscular junction. And as we see here on the board, the whole idea is the action potential boop, 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 travels down the axon, and then once it reaches the end, the synapse, it releases the vesicles with a specific neurotransmitter. There was a question that I had about, how do we know when the axon has an action potential traveling through it and when it does not? And we can see that as the action potential is kind of signified by these yellow lightning bolt arrows. Resting potential is when sodium is outside the cell and when potassium is inside the cell. When the action potential is coming, that's when the sodium and potassium are swapping places in the depolarization and repolarization steps. Now that the action potential has gone by, the resting conditions are being reestablished, back to sodium on the outside, potassium on the inside. And this cleft here, that is called the synaptic cleft. And it's very, very small, so that it can only be visualized with electron microscopy. So even though we have this space here and it looks rather large, It's actually very, very small. Let's take a look at it. So if we zoom in here on the synapse of an electron microscopy here, they do look very different from the nice and neat models we see in the textbook. Actually, let me make it a little larger here for us. So that space right there in between is the synaptic junction, the synaptic cleft. And the reason that there's all this stuff inside there is the neuron is a cell. And so there are some mitochondria to provide energy. And then notice all these little kind of clusters here. Those are the vesicles. Alright, what was the last thing I said before the camera was not working anymore? Okay, very brief pause. Alright, so we were simply saying that the action potentials flow from neuron to neuron. The axons connect with the dendrites of the postsynaptic neuron. So this pre and post, that's what we see here up on the screen. Alright. And just like the neuromuscular junction, these neurons diffuse across this very, very small space. They chemically bind to these receptors. The receptor allows sodium to enter the postsynaptic neuron, and we understand that when sodium goes in, that's the beginning of the action potential, right? So just like the depolarization, sodium going in signals the start of the action potential, and that is how the neurons keep passing the signal from one to another. Any questions about the sodium here before we move up to this calcium? Okay, and so remember that in the neuromuscular junction we also saw how a second action potential travels and triggers calcium ions to enter. So the action potential here, as it goes bloop, 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 as it travels down the axon like this, once it hits the end, it allows the it allows the calcium ions to enter in. So this is a very similar sort of setup as the neuromuscular junction, where the second action potential traveling down the sarcomere enables the sarcoplasmic reticulum to release the calcium. Except in this time, the calcium is not in any kind of sarcoplasmic reticulum. We can see here the calcium's kind of just floating around outside the synaptic terminal. Now, when this calcium comes in, that is the signal to let the synaptic vesicles go down and release their contents. So you have a lot of activity going on here. The action potential triggers the calcium, which triggers the vesicles to release the neurotransmitters. The neurotransmitters have to diffuse across the synaptic cleft, bind to the receptors, which then open the gates and allow sodium to come and start the next action potential. I know that's a lot to take in right now. Can we clarify anything about figure 7.15? Alrighty. So as you saw in your reading, we scoot here on page 222, that there are inhibitory synapses and there are excitatory chemical synapses. So there are synapses that stop information from passing to the next neuron. So this is a very odd thought. Think about this. We literally have in our body, some nerves are working to prevent information from reaching our brain. So what we actually perceive may not be what actually is, because we're not receiving everything from our nerve cells. Isn't that an odd thought? So like when we have various stimuli coming in, our body may be selecting which of those we perceive. So have you ever, I don't know, there's like instances where there is something, some sort of stimulus happening to you, but you ignore it, or you don't feel it until somebody points it out to you. Or if anyone has ever said, like, what's that sound? And you're like, what sound? And someone has to describe it to you, and then you listen, like, oh yeah, I never listened to that before. And sometimes you may have seen it too with like light or with touch, things like that. Yes, that would be nasty indeed. Your dad does that to you with annoying noises? Oh my. Yeah, so we will be looking at, or let's take a look here in the textbook and the examples that they give. So just like Gracie's example here. So in order for us to concentrate, oh come on, camera, there we go. So all the information here, if we listen to all the noises, the nervous system has gatekeepers to keep out all the information that we do not need. So the difference between the neuromuscular junction and the neuron-to-neuron synapse is that the flow of information is regulated from one neuron to the next. So an important point about these synapses is they make sure that our brain does not get overwhelmed with information. Now I know that there are some superheroes like Daredevil or Superman who have super senses, and the super senses allow them to do all kinds of things, but if your senses are like too much, then you have the disadvantage of being overwhelmed by the amount of information. Yeah, so I'm not sure if you saw that. I'm not sure what the movie was, but it was like the most, or maybe it was called Man of Steel or something like that. But I think it was there when like the evil Kryptonians come and invade the Earth, and they're trying to change it into Krypton, yada yada. And Henry Cavill as Superman, like the evil Kryptonians, when they get their mask ripped off or something, they're unused. So that with all their super senses, they actually get blinded by the high amount of sounds and lights on planet Earth. I'm not sure if you remember that scene, if you saw that movie. But it's sort of the same idea. We don't want to be overwhelmed by all the information that our bodies have. So the world around us, in all its information it has, our brains don't actually perceive. So can you trust your senses? Yes and no at the same time, because your body automatically regulates how much information is flowing through the neurons. Yeah, usually you should. And so one last example of this idea that our neurons are regulating our senses is, have you ever had, maybe driving along the road, or you're in the dark and somebody shines a bright light, and you're like, ah! Your nerves from your eyes, they're carrying less information. They're probably recognizing that that's too much of a stimulus. So even though we perceived a really bright light, it probably wasn't as bright as it actually was, because our body is actively working to try and prevent damage to the brain, to prevent the overload of information. So it's cutting down on that stimulus of the really bright light. Okay, let's take a look now that we've gotten some understanding of how the chemical synapse works. Why is cocaine so addictive, and what exactly is it doing to the brain? So here, what cocaine does is it blocks the dopamine receptor. So there are certain receptors in the body for various activities. So let's see, is there a bigger version? No. Okay, let's do this. Why is it so tiny? Let's try one more time. All right. So here, cocaine is represented by this little white dot right here. And ordinarily, this little white dot is not in the body. When there is a release of the neurotransmitters here, dopamine is a neurotransmitter that sends pleasurable signals. So, if you get that nice warm feeling after somebody hugs you, or you're really happy, dopamine is one of those neurotransmitters that is going through the body to cause that. And dopamine is one of many important neurotransmitters in the body. And notice that when cocaine is in the body, it blocks the ability of the dopamine receptors to go back into the presynaptic terminal. So notice this little white dot here, it plugs up that dopamine transporter. So normally, the nerve releases the dopamine, it does its job, and then it cycles back in. But what cocaine does is it blocks it. So it's like it keeps the nozzle turned on, as it were. That dopamine just keeps staying in the synaptic cleft, and that is why the drug makes people feel so high. So when somebody is getting high, that means that the amount of dopamine neurotransmitter is actually more than the natural level, because the cocaine is blocking your body from putting the dopamine back into the presynaptic terminal. And so the dopamine is just there continually stimulating that the receptors on the postsynaptic terminal. Any questions there? All right, so this is that euphoria of well-being. And so this feeling, it influences the brain to such an extent where it sort of tells the brain that, ooh, this is good. So the brain, in its reward center, the addictive nature of these types of drugs is because they cause this positive feeling, and so the brain gets reinforced to continue the use. And so there's not just like yes, it's addictive because once you like it, you want more and more and more. It's actually changing the brain and the brain chemistry. All right, let's take a look at this one here in the epidemiology. How much would you say till the brain dies? It can appear from person to person. But it's the stimulant there known to make users feel energetic, happy, talkative, etc. However, if you stop the message, right, so if you go back here to the image and you stop the cocaine, then the body will act as it normally does and decrease the level of dopamine. So if the brain receives a level of dopamine that it's not used to, you can have those negative symptoms. Let's see here. And so if we look at brain scans like this, there's a low frontal metabolism. That could be attributed to the loss of control seen in addiction. So when we study the brain later in the year, the frontal lobe is often the area for decision making. So notice that after 3 months, there's less activity going on in this space than there was before. Or I'm not sure, maybe this is the front and that's the back, but I have the arrows are pointed here. So the low decision making there may be why people, if they're addicted to any substance, whether it's cocaine or something else, is causing them to have lower brain activity. So it's often a very deadly irony that anything that is addictive, we think of it as stimulating, but actually it's lowering your brain's activity because it's just getting used to this this continued stimulant. So there's the activity of a normal subject compared to 10 days post or 100 days post. And, let's see, one last portion here. Unfortunately, some painkillers can also be addictive. Let's see, with cigarettes, I have not studied this myself because I know that some people are able to wean themselves off it by taking less and less because I know there's all the rage with those e-cigarettes or vaping, etc. So I haven't studied it enough. So if there are opioid painkillers, the opioid painkillers here, what they do is they have effects similar to heroin or morphine. And so what these do here is they capitalize on the body's natural pain relief system by binding to special pain receptors on cells in the brain and other parts of the body. So instead of blocking the recycling avenue for dopamine, what they do is if the body is in pain and there's a pain neurotransmitter, the drug binds to the receptor, which prevents the neurotransmitter from sending its message. So think of it like somebody glued the front of somebody's mailbox shut. Then the mailbox can't open to receive the letter. And so like the message is like, hi, I'm trying to give you a letter that says you're in pain, but your mailbox is shut, so you can't get the message. I mean, the analogy breaks down a little bit because then you can just like stick it in the door, but it's something similar. So stopping here can cause withdrawal symptoms like shakiness, vomiting, or diarrhea. And so lots of drugs, whether it's addictive drugs or opioids, they target the neuromuscular junction. Or excuse me, they target the synaptic cleft, that chemical synapse, because that's the kind of the center, it's kind of the heart of where messages are sent throughout our body. So a lot goes on here in this particular section at the synapse, which cause effects throughout the entire body, because this is how messages get sent through the nervous system. All right, any questions so far? Okay, let's switch to our sharing mode here so that we can get some more animations of the synaptic cleft here for us. All right, we're going to take advantage of the test. All right, that's working.
knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole...
He talks really fast, so I'm going to put him on 0.75 speed.
whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited.
So this is a very important point. Oh, his voice, do you want it to be normal? I can put it back to normal. It's a little fast. Okie doke. So the different types of neurotransmitters here they literally affect the type of signal.
The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area.
Alright, so please look at the bottom of page 225. Our textbook takes a long while to make this point, but at the bottom of page 225, in the second to last paragraph, signals only occur when there is enough excitatory signals. So once again, it depends on the sum of the excitations. So it's a little more complex than just, kind of one little stimuli from one location. It depends on how many action potentials. You can see in page 225, and let me just get my camera here to point out this wording for us. Excitatory synapses then do not, this is on page 225 in about the second to last paragraph. So excitatory synapses then do not just transmit all action potentials, they facilitate the transmission of action potentials, but it takes several action potentials on the presynaptic side of the synapse to stimulate just one action potential on the other side. And this is why we call the synapse a gatekeeper. So therefore, many action potentials can be like pew, 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 in order to get one action potential to go shoo, down on this side. So it's not a one-to-one ratio. There are many action potentials going along the side here. And that's the point that the video is currently making, which our textbook had this long, dry information piece about.
Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters. And sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets three major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize mood by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really, really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate, and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober...
And so, one of the main takeaways from here is that so many feelings can be triggered by these neurotransmitters. And this is a very interesting philosophical question. This, everything that we covered about cocaine influencing the body, or dopamine, serotonin, norepinephrine, this is why some people argue that human beings are no more than just a big bunch of meat influenced by different chemicals. So I personally have not met anyone who argues this, but there are some people who argue philosophically that human beings are just meat, meat and all that's going on in the brain. There is no such thing as love. There is no such thing as feelings, etc. It's just molecules moving around in the synapse. So, it is easy to see why this is the case, because when you look at the neurons, we can see how these influence our mood so much. And this also is an interesting question here, because if you take an antidepressant, I'm just like hypothetically, if someone takes an antidepressant, is that the drug that's causing them to be that way? Or is that actually them without the depressant? And this is a very interesting philosophical question where we understand that there is a chemical mechanism in the body to make us respond a certain way. And for various psychiatric disorders like depression or bipolar, or I haven't studied this much, there is a chemical side. There could be a mental side, like the person is willing this. There could also be, and from our perspective as Christians, there is a spiritual side. So I want to raise these topics because when we look at the chemical synapse, this is the heart of what influences our body so much. And we can kind of just like, yes, it's anatomy and physiology. But there are some interesting questions here with, what does it mean to be a human? And are these little molecules just like controlling us? Well, in one sense they are, because when I hug my wife, I get a release of serotonin. It's that happy molecule. So, is that emotion just serotonin binding to my neurotransmitter, or is that emotion the love that a wife has for her husband or a husband has for her wife? Is that all it is? It's just a chemical bonding to a receptor. Or, as Christians, we would say, no, it's more than just the chemical aspect. There is a spiritual aspect of this as well. And as Christians, we reject the fully chemical argument that, we're just a bunch of chemical reactions happening in the brain together. Other comments before we keep going here through the video? Excellent question. And so I have a friend who also is wondering the same thing. So my friend proposes that, because you feel anger, then that releases the chemicals, instead of, oh, the chemicals happened and made me angry. So I would have to study it a little more, like the will, or our heart, our desires. It's not something that you can just perform surgery on and be like, oh, there it is, there's the will. It's something very strange, because as Christians we understand that we are both physical, right? But we're also spiritual beings. So as Meredith proposes, I think that it is more along the lines of a biblical view of humanity, that our spirit may control the release of chemicals. But then it gets a little fuzzy, because like depression and other psychiatric disorders can be real, even though people may get the wrong diagnosis sometimes. And in that case, is there a physical aspect sometimes to your depression, rather than just being all in your head? Sometimes people do need to have their chemistry rebalanced, and it's a helpful portion. Sometimes it could be all in your brain. So it's hard, and we would have to kind of take it on a case-by-case basis. Yes, Meredith, I tend to agree, although I've never treated depression myself. So I would probably defer to biblical counselors on that. Right. Okay, so I will hide my camera here and we'll finish the rest of the video. Thank you for your thoughts, Grace and Meredith.
State, you got all these neurotransmitters doing their thing in your body, but once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors, so it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong, and when it comes to your synapses, that, my friends, is what wrong looks like. In their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science. Today you learned how electrical synapses use ion currents over gap junctions to
All right, so a good animation to go along with what we had talked about, and I just want to bring us through pages 224 and 225 because those were very, very dense. So, this from page 224, 225, and 226, the main point here is that it takes several come on, camera. It takes several action potentials on the presynaptic side of the synapse to stimulate just one action potential on the other side. As you go over here, this inhibitory synapse and stuff, we have to realize that a synapse is neither excitatory or inhibitory. Or excuse me, a synapse is either excitatory or inhibitory. It cannot be both. And so the difference between the two is the receptors. So the basic idea between excitatory or inhibitory means that it opens sodium channels. So if you open sodium channels on the postsynaptic membrane, or I'm sorry, I called potassium sodium. So if you open potassium channels on the postsynaptic membrane, that's an inhibitor. Because if you think about this, if you're opening the potassium channels, that means the potassium will go out. And we understand from this graph that whenever you are decreasing the chances of a signal, you're making it polarized. So in an inhibitory synapse, where did that diagram go? Here it is. By releasing the potassium outside, you're actually making the axon more polarized. And that means it's even more at a resting potential. If the receptors open sodium channels, it's an excitatory synapse, because when we open sodium, that allows the sodium in, depolarizing it and making it more likely to carry the signal. And that's basically the only important difference between the two. So would you like me to go through that again? Okay, let me get that image back on the board that we saw before about the action potential, and maybe this will help, and we have tomorrow to go over it as well. All right, so Meredith asking is when sodium increases and potassium decreases, the positive increase causes the action potential? Good question. So when the sodium, it's not really an increase or decrease, it's they're swapping places on page 214. So it is when the sodium enters into the cell and the potassium leaves the cell that the action potential is at its highest. So if we combine steps B and C, within the axon, let's see, when sodium and potassium decrease. Yes, that's correct, Meredith. So what Meredith says here is at this particular point in green, here on the picture. So notice in green on the picture, the highest spike in the potential difference happens when sodium is all inside and potassium is leaving. So that's the height of the action potential. And then the repolarization is when the sodium-potassium pump begins putting things back to normal so that we return to the resting potential. And we see that drop. We see that repolarization step in the graph here on the side. All right, so it sounds like folks are still mulling over it. It takes you some time to go through this because it is very dense. We will practice that tomorrow and finish up Chapter 7 so that we can take the test and finish Chapter 7 before Thanksgiving break. Class is dismissed. Have a good